Last August, Michigan State University chemist Dantus and Elad Harel received a $1.3 million grant from the Keck Foundation to start a new revolution in the way we use optical microscopes to understand the living world. The grant, awarded by Keck Foundation's Science and Engineering program, encourages creativity by rewarding transformative projects that other funding agencies might see as too ambitious or risky. The grant also marks the first time that scientists at MSU have claimed the award. Dantus and Harel will team up to use laser light to record movies of biological processes at an unprecedented resolution. The research will push the limits on what light-based or optical microscopes can see, which will ultimately bring benefits to a range of scientific and engineering fields. Hello and welcome to the podcast. Can the two of you talk a bit about how you began collaborating at MSU?
We're always interested in collaborating because even though we're both doing ultra-fast spectroscopy, we really came at it from really different directions. And I think there was a lot of complementary capabilities and ideas that were coming out of our group. So, ultra-fast science in general, which is the study of very, very fast phenomena that basically can only be studied using really short pulse lasers, is a really broad field. It didn't start out that way. Marcus was actually at the very early stages of the inception of ultra-fast science, many years ago. But it's really grown and touched upon so many different areas and different applications. And so that's actually what made it and always drove me and attracted me to the field in general, where all the different areas of chemistry and physics and material science and so on, biology, that ultrafast touched. And so we had always been kind of thinking about collaborating, but when this idea for the Keck Foundation and the call for the Keck Foundation came out to develop new methods in imaging and especially tackling living systems at very, very high spatial resolutions, well beyond what could be achieved optically, it was just it made a lot of sense for us to combine our expertise. And so it was kind of a no-brainer, I would say, like we should go after this together, because I think individually we wouldn't have really been able to do it, because there are a lot of technical challenges in this kind of project. And this is testament to the fact that people have been trying this for a very long time. Basically, from the inception of microscopy, people have been trying to improve the resolution, and they kind of hit a hard limit when it was recognized that one was limited by diffraction and the wavelength of the light being used and so on. And so I think it made a lot of sense for us to work together. And we brought different, as I mentioned, we brought different kind of expertise. Marcos really brought the expertise of the light sources and the really challenging needs to shape these light sources in certain ways, spatially and spectrally and temporally and so on. And we brought some of the ideas, especially from my background in magnetic resonance imaging, bringing those ideas and relating them to what had and had not been done in the field of optics. So it was kind of a unique opportunity based on our backgrounds and also based on our current interests. And it fit together perfectly for tackling this challenge, one that I think both of us have been thinking about for quite some time from really different perspectives.
The Keck Foundation is well known for supporting risky, ambitious, and highly impactful projects. In your case, you proposed to invent a laser-based system that might allow us to finally record and observe living systems in motion. Was there a particular moment that you can recall that led you to begin researching this idea, or was it a concept you already had in mind?
Observing cells as they are, living has been a challenge for a very long time, and there have been many methods that have been developed to first get depth resolution, so that you can sort of saying like slice the cell and look with the depth resolution and spatial resolution in general. Unfortunately, those methods are limited by what's known the diffraction limit. So on the order of half of a wavelength, or let's say 300 nanometers, give or take. And there have been methods that were developed for super resolution. In fact, they received the Nobel Prize, where you are able to achieve super resolution with a laser source. However, those methods are very slow. And so if you wanted to observe a cell, a living organism where things are moving, then they're not widely applicable. I don't want to say impossible. At the same time, Elad had some ideas on how to change the equation, so to speak, and convert spatial resolution into the time domain. And so we had a brief conversation, and as this idea, as the call for the Keck proposal came out, that's when we said, aha, why don't we write a very compelling proposal that combines both of our expertise? And if you want to add more detail, Elad, on that.
As I mentioned before, I came from a kind of different background, where in my PhD I worked on nuclear magnetic resonance and magnetic resonance imaging. So, these are the scanners that you go into when you go to a hospital, and they'll scan your brain, or they'll scan your heart, or scan other organs of your body. And one of the things I always thought about when I entered the field of optics was this idea of, what does the diffraction limit really mean? And where does it come from? We kind of take it as a given, like it's a law of nature. But really, the diffraction limit is, while it holds, it's a limit, it holds because of the way the experiments are conducted, basically. That, you're trying to make a spatial measurement, it makes sense to, if you're trying to measure 2 points in space, it makes sense to make a spatial measurement. However, in magnetic resonance imaging, you're getting spatial information without making any spatial measurement at all. People probably don't realize that when they go into an MRI scanner. But what you're really doing is you're encoding different spins. These are what you detect at the end of the day. You're encoding them using what's called a magnetic field gradient. You're basically letting each spin in different parts of your brain, or whatever the sample may be, experience a different frequency. And because that mapping between space and frequency, you can recover basically the kind of tomography, if you will. You can recover where that spin originated from. So in magnetic resonance imaging, you're not taking an image like you do with a camera. And the most important part of that is that there is no diffraction limit in magnetic resonance imaging. You're not limited at all by the wavelength of a radio frequency wave, which is on the order of meters. Otherwise, if you were, you wouldn't be able to see any resolution inside of something like the brain. So I had thought about that for a long time. And in my postdoc, I kind of used some of those ideas, but in the optical regime, not to do imaging experiments, but to do spectroscopy experiments. So when the Keck Foundation call came out, I had also been thinking about, well, could you do this? Could you make an analog of the MRI optically? Can you make a purely optical analog of that? And that kind of made a lot of sense for kind of achieving the goals of the Keck Foundation. Now, the challenge, though, was how do you make the equivalent kind of gradient, which is what you use in MRI, how do you do that optically? And I think that's where, discussions with Marcos, we thought a lot about how to do that. And what would that look like? And as Marcos mentioned, how do you get the speed, right? Because MRI scanners are not that fast. If you've ever been in one, you have to sit there for 20 or 30 minutes. And, sometimes longer. So they're not terribly comfortable. That's not really going to work when you have cells that are dividing, or if you have some kind of biological process you want to watch, which happens on a much faster timescale. So there were then technical challenges of how do we do this at the right speed, and how do we do this using very different detection modalities, and the physics, frankly, is also very different. So that's where that's really the kind of, I wouldn't say there was like this aha moment necessarily, but there were this kind of hopeful feeling that we could overcome that the actual technology was there to overcome these challenges. And a lot of the technology that was needed, in the MRI scanner, you can kind of type in pulse sequences and do all this kind of manipulation so-called pulse shaping just on a computer based on really well-designed RF, well-developed, I should say, RF technology that's been around in telecommunications and all kinds of other communication systems. That technology didn't exist 20 or 30 years ago in the optical domain. And a lot of Marcus's work actually developed that kind of computer-controlled pulse shaping. So it was a technological barrier, not only a conceptual barrier, but a technological barrier that I thought, we both felt like, wow, the technology is probably available, right? It exists. It's not some hypothetical device that we have to dream up. It's there. And so that's where I think that was kind of the aha moment, if you will. Like, the technology is there, it exists, but it has to be put together. And that's not a trivial thing to do, but it's a possible thing to do. And that's what allowed us to put forth a proposal.
What sorts of challenges have you encountered in this type of research, and have the solutions or workarounds you've discovered created new research directions or led to any surprising insights?
It's really wonderful when you get this kind of award and you have a direction and you go in the lab and you start testing, right? And if everything works immediately, it means it wasn't challenging enough, right? Someone must have done it already. So you start testing and you start finding the limitations of everything. So, for example, we have made a tremendous amount of progress that I can talk about maybe a little later. But one of the challenges that we're finding is when you're looking at things that are smaller than the wavelength of light. So if the width of a hair is about 100 microns or 100 millionths of a meter, and the wavelength is, let's say, in the visible around 600 nanometers. So that's 600, billionths of a meter. We're talking about imaging things that are, 20, 30 times smaller than a wavelength. And so that's like 20 nanometers. And so any vibration, even if you talk right next to the microscope, everything is vibrating. So one of the things is wind currents, vibrations, temperature stability, all these things. And then we amazingly are at the moment having problems that the optics that we're using, when they are polished, they're not polished perfectly enough. And so that is kind of what is limiting our progress in my laboratory. But it's very exciting to be there. After, I think that if you would have asked us this question after the first month, we would have said, oh my gosh, it's not very clear what's going to solve all these problems. And now, about, let's say, approximately a year and a month, we have very exciting research that Elad can tell you in his lab. I can tell you about my lab.
This is really the nature of research, right? You go in and you try things, and you're discouraged sometimes because you feel, wow, there's a lot of things I didn't think about. At the same time, that's really what creates new ideas, right? If you were never challenged, you would never come up with a solution. So I think that's kind of one of the really interesting things. And I think foundations like Keck know this, right? Like, they know that what you write down on paper may not work exactly the way you envision, and that's okay. They know you're going to come up with creative solutions to that, and that may go off in an interesting and unpredictable direction. I think that's kind of what's happened here. Marcos, the stuff, I don't want to speak for what he's doing, but the stuff that he was doing kind of probably wouldn't have happened without Keck, right? It was like the challenge came there. I was like, wait, how do I tackle that? Okay, the new ideas came out of that. And we, even while we were writing the Keck proposal, we had tried some of the things in our lab, and we realized pretty quickly that there was an opportunity, and we didn't even incorporate it into the proposal at that point, but we had seen an opportunity to incorporate not just the imaging side of this, which was kind of what the proposal was about, like, how do we see things, right, at this resolution, but what we wanted to incorporate was the spectroscopy side. How do we really get detailed information about what's happening, what is happening structurally, chemically, right? What are the interactions at this kind of molecular and nano scale? And we realize that that's equally important. It's not just about what you can see; it's also about what the things you can't see. It's not, you can't see everything. A lot of these interactions are not visible, even if you had all the resolution in the world. So that led us to think a lot about a totally different direction where we could do spectroscopy on these living systems, and where the spectral signatures were really, really unique, even for these really small biological entities on the order of these, tens of nanometers. And so that kind of spawned a whole research direction. And I think then I hope happens at the end of this is that the spatial part of the problem that Marcos has tackled, combined with the spectral part of the problem that we have tackled, can now be merged together. Because ultimately these problems are spatial, spectral, temporal, right? It's like you have to see the whole thing. It's like watching a movie. You need the spatial parts to see something. You need to see the screen, right? What's happening? How are things moving? You need the time part to see things changing. Otherwise, it's a very boring movie, just a picture. And you need the spectral part, and in that case would be like the colors in the movie, the red, green, and blue. They give you information that you may not see in black and white. And so it's kind of combining all of that is what gives the richness to the story. And the same thing in molecular systems and biological systems. We need to see all of those aspects of it to really get a full picture of what's happening. If we just watch one thing and ignore the rest, we're going to have a really hard time understanding what the interactions are, what these processes are, what the mechanisms are, and so on. So, in some ways that's the challenge, but I think the fact that we've very early on identified what the most important aspects of that were is kind of what has now given additional life and additional sides to the story. So beyond what we had proposed in the original Keck proposal.
Could you speak about the different ways your research might be applied now and in the future? And in doing so, is there one specific application that's especially exciting for you?
Elad and I have been people that have developed a number of laser systems and methodologies and so on. But we also should give credit to the people that are in the lab. We tell them the crazy ideas and then they try and it's discussions together that gets the things moving along. In particular, I have a fantastic scientist from Russia that has been working on this microscope. His name is Sergei Anisic, and he's doing a fantastic job. In fact, we can now see features that are 30 times smaller than the wavelength of light, and we're very, very excited. Our system is pretty slow right now, so we could not see something in motion. So it takes maybe 10 seconds to get a full picture, which may seem fast, but not for something that is living. For something that is living, you want to take several pictures per second. But we have already ordered the parts so that we can go much, much faster. Our goal is to do 100 pictures per second once everything is working. And we are still we have not solved all the problems. We are working on it. But right now, our system could already be used for things that are used in let's say, microchips and and electronics, where the part can stop and you can take a picture, and if it takes a few seconds, it's okay because it's not living. So we can image non-living systems or things that move on the 10-second time scale. But hopefully in the future we will be able to do faster. And then Elad has some very, very exciting work that not only will tell you there's a feature there, but what is that feature and identify it. And I want Elad to elaborate on that if you want.
We've moved into this kind of direction where as I mentioned before, we're trying to use spectroscopy to identify features or collective features in different systems So what I mean by that these are, if you think about, I like to use the analogy of sound because it's actually a really clear analogy. We know that every material has a unique sound, right? We tap a piece of glass, it's going to sound different than a piece of wood or a piece of metal or a drywall or anything else. Molecules are kind of similar in that way. If you hit them hard enough, they will produce a sound. Essentially, they will start to vibrate. And people have studied local vibrations for a very long time, like bonds moving and vibrating and so on. But what's been far less studied are kind of global vibrations. These are motions that all the atoms in the system are collectively vibrating together. And what we're kind of discovering is this collective excitation, these collective sounds, if you will, these different modes are unique, right? Every species sounds slightly different. We can resolve that. So, you can think of lots of different types of applications for this kind of work. You could imagine there could be applications in medicine for distinguishing different, or biology for distinguishing different biomolecules without labeling them. And that's a big aspect of this. This is totally unlabeled. So you don't have to do a lot of workup, and you don't have to figure out where all these different sites are. So that can be really expensive and very slow. This could be used for things like forensics. It could be used for counterfeiting. It could be used for, detecting explosives. It could be used for lots of sensing applications where you want to identify and distinguish two very similar objects. So, there's lots and lots of different applications. I think that we're beginning to explore, and we're really excited about that. And then there's, I think ultimately this will hopefully have an impact on just our fundamental understanding of molecular interactions, right? And how those are changing and into highly dynamic systems. So I think there's just many opportunities to interface to many different disciplines outside of chemistry, to physics, material science, biology. And then we've also brought in a lot of challenges on the computational side, right? How do we simulate these collective modes? There's really very few people doing that. It's a very challenging problem because you have thousands of atoms, maybe tens of thousands of atoms, and you have to really account for all of them. Then there's computational tools about how to analyze this data. How do you look at this data? What does it mean? How do you extract the features from this data? So it's kind of opened up for us a lot of different collaborations, a lot of different interesting challenges and overlap and interfaces with many different fields.
Given that you're the first MSU scientist to receive a Keck Award, can you talk about the unique opportunities this honor provides and what makes this ongoing project unique compared to others?
Well, I'll start with the saying that it's a tremendous honor to get an award like this. We are scientists, and we're always, our brains are always working. And I'm sure it's the same with Elad. You might be sleeping, you might be in the shower, you might be running or doing whatever, and all of a sudden it's like, oh, there's this great idea. Or you hear about challenges, and sometimes it's a new challenge, but many times, like this time, this is an old challenge. We've always wanted to see the living working cell. And even though we have techniques such as electron microscopies or using X-rays for imaging, those would kill the cell. And so you need to image with super high resolution and speed something that is alive. So you need to, our idea was, starting with Elad, can we use just regular visible light? And the challenge was, okay, but it has a very long wavelength compared to the items that we wanted to see. But when you get an award like this, it's a chance to think completely outside of the box and to be able to try and fail, but you have a runway of funds, so you can gather the smartest people and and think very hard, and then you work very hard in the lab, and then as you fail, you take lists of, okay, this failed because of A, B, C, D. How are we going to solve those? And so that's the thinking hard part. And then you go back to the lab and you work hard again, and you fail again, but you know it's gonna, you're making progress. And that's the key. You need to, so an award like this gives you a runway, so to speak, so that you can have very smart people working with you, and you can think and push yourself to areas that nobody else has tried, because otherwise the challenge would have been solved, and there's no challenge anymore. And it's this idea of being able to fail and remaining optimistic, because you have the time and you have the funds that you can kind of have a version, the next version, the 2.0 and the 3.0, and the, and it's going to take 10, 20 iterations before you start finding exactly what is working. And so it's really fantastic. It's one of the best things that can happen to a creative scientist, because you can try and remain optimistic that well, if this fails, we will try something else, and if we find that we need something 10 times more accurate, we can afford it, and get it in the lab and then try again.
Yeah, I think that so I agree with everything Marco said. I think the unique aspects, I mean, obviously it was an honor to get the Keck Foundation grant, because it was great great to have it here at MSU. It was a real honor that it was the first one that they had received. But I think, for me, and probably certainly for Marcos, I would guess, the greatest thing about it is that this is like unrestricted funds to explore, any ideas that would really not be fundable, easily funded in any other way. And so there are, nowadays, there are just not very many opportunities to get these kind of resources without having a significant amount of preliminary results, and a track record. I mean, we have a track record, but a track record of this specific project. And I think that just doesn't happen often. And having the freedom to kind of every scientist's dream is just to have the freedom to do whatever they want, and explore all kinds of crazy ideas. And oftentimes really good things come out of that. Not what you expect, but really good things. I think Keck knows that. They know that this is basically funds to leverage, right? This is what they want, is they know it doesn't end. They hope and they screen for, in fact, projects that don't just end at the end of the grant period, right? The whole purpose here is that they're providing effectively seed money to seed an idea, which will grow and become something, and which will then be amenable to other funding sources. So something that can have longevity of 20 years, 30 years, and maybe more, right? Maybe it gets taken on by postdocs and graduate students and other groups, and so on and so forth. This is how fields get developed, right? Someone took a really crazy idea, they made it work, and other people picked it up, and it just developed and developed. And actually, that's the way good science happens, because you can't do things in isolation. You have to bring in the whole community of scientists to develop these methods, apply them, and solve real problems. So I think the most exciting part of this for me is just all the tentacles and all the connections that this can potentially create. And the fact that it's going to lead to, I'm almost certain it's going to lead to new research directions for both our groups. And it's going to pay itself forward many, many times over to future generations. And so that's why it's really important that we don't squander the opportunity, and that we really make something of it and utilize that opportunity, so that, people can look back however many years and say, look, that started from a Keck Foundation grant. And I think there are lots of examples of that if you look at their previous track record and of other private foundations where, really important ideas kind of came out of it. But they did take many, many years. It didn't take 2.5 or 3 years to see something as complex as this all the way through. It takes many iterations for it to finally get to the point and maturity where it can be used, say, routinely by a biologist. So I think that's just a pretty exciting and unique opportunity. They don't come along very often. So yeah, I'm certainly like super grateful that we had that opportunity.
Is there anything we haven't touched on that you would like to share with our audience?
Well, I guess I would also, I mean, the one other thing I would add is, the support of MSU was really important here. So we had the foundation's office and all the different entities that were involved in this, up to the VPR's office, where, they really just were ready and able and willing to help throughout the whole process. And that was really important. So to have the university's backing and support and determination to see this through and to make this work and to believe in us was super important. I don't think it would have happened without that support. So it's kind of shows that, the power of an institution, right? Institutions build the labs, right? They support the students, they support and help bring startup funds and help support with bridge grants. And there's all kinds of different ways in which an institution like MSU helps in making this actually happen. It doesn't happen in a vacuum, right? We don't have just labs that are unconnected. We're all part of this community. And I think that is absolutely critical, that the university's involvement all the way through from what we do all the way up into different parts of the administration. So that was really key. And I really did appreciate that.
I would like to tackle another side of this Keck Award. We talked a little bit about it, but I think it's extremely important. My PhD advisor was Ahmed Zuel, who got the Nobel Prize in Chemistry in 1999. And he was very adamant that we shouldn't forget about what he called curiosity-based research. And to just give an example, when Albert Einstein discovered, and started writing about relativity, he didn't sit down and said, I am going to solve this problem, which will lead to all sorts of, everything that we know now. He was just intrigued by an experimental result. And based on that experimental result, he said, well, if that is true, then space and time are not exactly what we think. And so our equations need to be modified. And so turns out when we were talking about the honor of getting one of these Keck Awards, is that if we apply to any of the federal agencies right now, not only do you need some preliminary results, but in many cases, they want to know, okay, but how is this going to be applied? And so sometimes you start with a challenge and you say, I'm going to solve this challenge. But there is the part of curiosity-based research. I am going to make an instrument, or I am going to use a new approach and look at an old problem and probably find things that nobody has observed. And maybe I'm not sure what I'm going to see or how, but it's that curiosity-based research that leads to breakthroughs. And breakthroughs, are exactly what changes the world, right? It's not the same to get 5% better efficiency at in some motor or something like that, than to completely change and say, well, now we're not using combustion anymore. And this motor is now, serves the purpose of a motor, but it's revolutionary. It's a breakthrough in terms of how it consumes energy, or maybe even generates energy. And so I think that that's something very, very important, and we mustn't forget. And I hope that every agency reserves some funds for this kind of thing, this curiosity-based research where, well, we're going to try to measure something new. We don't know exactly what we're going to find, but when we find it, I'm sure we will learn a whole new universe, right? And in a sense, that's kind of what we're shooting for in this proposal. When we have the super resolution using visible light, and we are able to look at a living system, we're going to find new things that maybe we only imagined before, or we could see indirectly by very cumbersome methods.
In closing, if people want to stay up to date with you and your research, what is the best way for them to do that?
A good way is to reach out to us, and we love to tell people more about what we're doing, or give people tours. We have websites that people can check out, but I think really the best way is to talk and to kind of try to explain these ideas, and really also get new ideas for people as well, and think about we actually shortly after the Keck Award, we got some emails about could you use that for doing this and this and that. I was like, I don't know, maybe. I never thought about that. That sounds like his idea, it just never occurred to me that people have been thinking about and have expertise in all kinds of interesting systems and applications. So I think communication is really important. That's how new ideas come about. And so I'd be more than happy to talk to people, and they can contact me via email.
Yeah, me as well. I don't tweet.
Or X now. What is it called? I don't know what's called.
Yeah, or X. Yeah, no, I find that any of those social medias would be taking me away from the lab. And what I like to do the most is to be thinking about science or working in the lab. And so, but yeah, I mean, we have websites, we have emails, and we can always set up appointments. And I like what Elad said, we can set up tours or whatnot. It is this communication with everybody that can take us to new ideas, right? If we're developing a tool, we don't know all the different applications that tool may have, and those would be very interesting to explore.
Well, this has been great. I'd like to thank both of you for joining me on today's show.